Hello, my name is Dr. Lisa Kueltzo, and I am going to be talking to you today about vaccine production, specifically the vaccine production program at the Vaccine Research Center at NIH. This talk is part of the Clinical and Translational Research Series for the 2021 Summer Seminar course, and it is a follow-up to two other talks, which hopefully you've already had a chance to view. The first is by Dr. Camille Lang, which talked about translational sciences at the VRC, providing an overview, and the second by Dr. Jeffrey Boyington, who talked about the basic design of influenza vaccines. The follow-up when we talk about translational science, moving from the bench, where Dr. Boyington's talk started, to actually the injectable clinical vaccine that can be given to a patient in a trial is production of that vaccine, and that's the subject of my talk today. You perhaps have seen this slide already, but this is the development cycle that is used at the Vaccine Research Center at the NIH. And as I said, Dr. Boynton talked about basic research. Dr. Lang gave an overview of this whole slide. And today, what I'm going to be talking about specifically is process development and a little bit about manufacture. So when talking about vaccine process development, I always feel it's important to let people know that it's a very interdisciplinary field. It applies theories and approaches from a variety of scientific disciplines, including molecular biology, biophysics, and thermodynamics, multiple areas of engineering, both at what I would consider general engineering principles as well as specifics of chemical and biochemical engineering. And our goal through vaccine process development is to develop a production process, analytical methods, and clinical formulations that result in consistently safe and efficacious vaccines through multiple production cycles. Now, the challenge here is ensuring we can get that consistent quality, safety, and efficacy when we move from a very small scale to a much larger scale. And here we start off with, in the basic research realm, our single vaccine in Eppendorf tube, moving up to a 2,000 liter bioreactor. And that's what we have at the VRC. You can see hidden there a person for scale in this lower half of the picture. But that can easily be 20,000 liters at commercial scale for production of large-scale vaccines of biotherapeutics, resulting in thousands to millions of doses. And here we see actually a vial of our VRC01 HIV monoclonal antibody, which is actively used in VRC trials. Now, before we start developing a process, we go through what's called a manufacturing and biophysical risk assessment, or an MBRA. This is an early screen of research material to look at risks for manufacturing and development. And this starts off with looking at an assessment of, is the candidate actually appropriate to develop it? And that's looking actually at the molecular properties of the molecule and looking at different areas where we know there are risks and highlighting areas that might be required for follow-up investigation. So if a risk is identified, the question is, how severe is that risk? Is that something that's going to completely impede development or something we think we can develop workarounds for and solutions for? Following that as feasibility assessment, assuming the candidate passes, we go into what's called a process feasibility assessment. That's looking at potentially different cell lines for growth of the material, looking at might be what standardized processes and analytical methods we already have. Are those going to be applicable? It gives us an idea of how the actual development is going to proceed. When we look at the categories of risks that we're trying to identify within the MBRA, we look at things like primary sequence liabilities, and so we'll perform a sequence analysis. We're looking for areas of potential cleavage, looking for deamidation hotspots. Biophysical characterization, there we're looking at structure-related liabilities, poor conformational stability, low thermodynamic stability. We look at colloidal stability as well. And here we're looking at, is it aggregation prone? Is there poor solubility if we need to generate a product where we have very high concentrations? Do we see something called liquid-liquid phase separation, which also indicates colloidal stability? All of these types of risks could result in even if we can generate a product, it perhaps not being stable for more than 6 months or a year, which causes a lot of problems down the line when we get into clinical trials, which can often take several years to complete. We will look at platform solubility, and there we're looking again, as I mentioned in the slide previously, how well this will perform in key process steps and platform analytical assays like size exclusion chromatography. We'll also look at what's called the target product profile feasibility. Target product profile is what we feel early on, and by we I mean the entire VRC, including the clinical trial groups, is going to be the best format for the drug to enable the clinical trial. Do we want a frozen drug, a lyophilized drug? What concentration will it be at? Is it going to be fine in a vial? Would it need to be in a pre-filled syringe? All of these are the target product profile, and there are different challenges developing that in each case. And then another area to look at sometimes is cell line equivalence, where we evaluate the risk using different types of cell lines or transfection processes during development and how that will affect the overall success of the development, as well as the timelines and any resource needs. So overall, if we're looking at the vaccine production process and looking at the scientific steps, we're starting off with DNA, we're moving on through various other steps in production, et cetera. And these can be divided into several categories, and I'm going to go through those in the next few slides. So the first is cell line development. That is where we are taking the sequence of the molecule, generating plasmids, cloning them into cells, and developing stable cell lines. Then those cell lines are moved into what's called upstream development or cell culture development, depending on the organization. And that's where we're scaling up from one small Eppendorf tube of cells to, say, 50 liters, 2,000 liters of cell culture. That cell culture needs to be purified. That moves us into purification, or also called downstream development. And at that point, there are multiple what we call unit operations in that area to basically separate our molecule of interest from a variety of other impurities and just, other substances produced from the cell culture. And then we move into formulation development. And there, the goal is to take that purified product and ensure that it does not change in terms of safety, efficacy, et cetera, over the course of not just moving into a vial, but several years of storage in freezers or refrigerators, both in the manufacturing storage facility as well as in the clinical sites themselves. So let's start off with cell line development. The goal of cell line development is to generate clonal cell lines and or stable pools for clinical material production. And so that starts with the plasmid, moves into transfection of cell lines. We often use CHO, which is Chinese hamster ovary cells. Then from that transfection, specific clonal colonies are picked and expanded into shake flasks. And the reason we would go with clonal cell lines is because that ensures that the protein produced, so this is specifically looking at protein-based vaccines, are going to be consistent over the entire production. The top clones are identified, and then those are transferred to upstream development for further scaling up, as well as a number of cell culture aspects, which I'll get into in a moment. One of the things that's often most different in looking at things from a lab scale to a manufacturing scale is the type of production yields we're looking for. And so if we have a stable pool for vaccines, often at the pool stage, we want up to a couple hundred mg per mil. When we get to clonal cell lines, definitely 100 mg per mil and greater, often 400, 500 mg per mil is wonderful. When we talk about monoclonal antibodies, which are administered at much higher concentrations and in much higher doses, a stable pool, we want a gram per liter of purified, a gram per liter of material expressed at the cell culture stage. For a clonal cell line, we're looking at greater than 3 grams per liter. It's an incredible amount of material, but it's important to realize that a dose you could definitely be receiving a couple of grams per dose, depending on exactly how the trial is constructed and really what the efficacy of the antibody is. And then the timeline for stable pools are much shorter, which is why we consider starting with those about 6 weeks, but a clonal cell line could take about 4 months to produce. This is I love this picture. So this is an instrument called a clone pix, and this is one of the instruments used to actually pick colonies in a very high throughput. And you can have thousands of colonies that are generated during this process. And what this instrument does is it automated in an automated fashion, seeds the colonies in a semi-solid media, and then will pick little plugs of that media and then transfer those to 96 well plates for further evaluation. And here you can see an example of some of the wells of the media that are examined, and the green colonies with the kind of larger halos would indicate highly producing cells we'd want to move forward with. So after we have those clones selected, we maybe have some lead clones, maybe 3 to 10. Those will move into upstream process development, where we're starting to look at cell culture attributes. And so during clone selection, the individual clones are all compared, and at that point, we're about a 15 milliliter scale. It can go up to 250 mils. And we actually use robotic systems, which are high throughput automated cell culture systems. So a wide variety of conditions can be evaluated to compare productivity, product quality, etc. At which point then we scale up to process development scale, which again, we start to get into a few liters, and their production pH, temperature are defined. We're looking at media feeds, etc. The next level of scale up, at this point, we have a lot of the cell culture production process defined, and we're starting to try to generate process representative material. At that point, at least at the vaccine production program, we scale up to 50 liters, and we regularly produce batches that have 50 liters of cell culture. This not only confirms the process is working as we continue to scale up and is consistent, but it also supplies material for toxicology studies as well as downstream formulation and analytical development. Finally, when this process is then transferred to what we call our GMP pilot plant, and that means that material is manufactured under good manufacturing practices, which is a regulatory category. We can scale up to anywhere from 250 to 2,000 liters at the NIH pilot plant. And as I mentioned before, it can be larger at other sites. Now, downstream process development. The goal of downstream is to separate our active target drug substance, circled here in the picture, from cell debris, byproducts, manufacturing process additives, product-related impurities such as aggregates, and that's all done by exploiting the various physical chemical properties of the target versus the contaminants to achieve these very specific separations. As I mentioned a number of slides ago, there's different what we call unit operations, and those are listed here in this diagram. And it starts off with aspects of, say, clarification, which is really just separating out the bulk cell debris, to UFDF. Now, UFDF is ultrafiltration diafiltration. It's often also called TFF because tangential flow filtration is the specific methodology most used. And there we're changing buffers, we're concentrating the material. And then there's usually at least one, if not 2 or 3 chromatography steps, and a couple steps here. You'll see viral inactivation and viral filtration. Those are very important because we need to inactivate any endogenous viruses that might have been present in the growth cell line to ensure patient safety. I talked a little bit about column chromatography. That is the workhorse of downstream development. And here's an example of what we talk about. We're talking about scaling up columns. So on the left, you have here basically a robotic screening system. We use that here at the VPP. And there you have small columns, 0.5 mil to 1 mil in size, and you can automatically screen 50, 100 different resins under different conditions to see where we're actually getting binding or where we're getting binding of our contaminants to eliminate those. And then once we have some leads, those get scaled up to, say, 50 mil, 5 mil to 50 mil columns on an FPLC. Many of you may have used one of these before. And then once we have a process locked, then it scales up to manufacturing. And again, here we have a picture from a manufacturing plant. And just for scale, you can see there are 2 very large columns, and these can be 5 liters up to 50 liters or more in size. And all of the white space you see there is actually column separation gel. So that's resin. Just to give an idea of the size we're talking about here. Now, formulation development, the goal is to start off with a pre-formulation drug or vaccine. And so this is what's come right out of the downstream process. We have some purified material. And then the formulation group will take a look at the pH, ionic strength, buffer salts, and a variety of stabilizing excipients and identify what the solution condition is that will be optimal for stabilizing that vaccine or drug in terms of its chemical and physical stability to maintain the structure and therefore the efficacy over the course of time we expect to use the drug under the conditions at which it's stored, whether it be frozen, refrigerated, room temperature, etc. So we start off with the pre-formulation drug vaccine. We look at pH and ionic strength. Those are huge factors. We then identify stabilizing excipients based on how it's going to be used and stored. Moving along to the point where we have some good and some bad conditions, as you can see there in the middle. I'm going to use red for bad, or rather less stable. And at that point, we'll take the conditions at which we are seeing the best stability in terms of the actual intrinsic properties of the molecule and look at what we call real time. So that's under anticipated storing conditions as well accelerated stability data. So accelerated there, we're talking about maybe higher temperatures or some other sorts of stress. And then from that, we can determine what we feel is going to be the best formulation for use in the clinic. And that basically formulation gets locked or selected. And then the process is transferred again to the manufacturing plant and also used to generate some data to support clinical trials to ensure that the formulation is going to be compatible with IV bags, syringes, etc. Now, one thing I haven't talked about very much through this process so far is analytical development. And that's because analytical development doesn't really occur as a step in the process as much as supports the entire development process. And this actually starts in basic research where there may be some clinical reagent evaluation all the way through the process of formulation development. And then, of course, GMP manufacturing and can extend again into clinical trial support. So the assay development area is responsible for actually ensuring that the clinical vaccine product meets all quality specifications. So there are assays needed for content, potency, identity, different types of impurities, safety, stability, etc. They also have to develop and generate assays for in-process content and quality. And that supports actually the production process to verify that we've met, say, the correct cell titers. Or perhaps the DNA has been removed to a sufficient level after specific chromatographic steps. They also, analytical development, because of the fact that they are the subject matter experts on the majority of support assays used for GMP manufacturing, are involved in a number of collaborative investigations. And this may be looking at how process changes affects vaccine quality, looking to see if current assays could be applied to novel vaccine modalities, and then definitely evaluating any adverse stability trends that may or may not occur over the lifetime of the product. In a bit of a nutshell, that was the production process overview. And I'd like to say that it's all simple and goes smoothly, but that's not a very interesting story. Not surprisingly, this is science, and unanticipated challenges can definitely arise. And I'm going to talk about a couple of the what I feel are the more interesting ones in the next couple of slides. First of all, though, it's important to remember, just like many things in life, things aren't as simple as they seem. I've presented everything so far as kind of this straight line process, but in real life, from plasmid to clinical vial, it's not a straight line. It doesn't happen that cell lines are finished and 100% guaranteed, and then we move to cell culture, and that's 100% done before we go to downstream. In reality, as soon as the cell line development reaches the point where we have some leads, they move into upstream, as I mentioned. And as soon as the upstream group is starting to generate some material, that immediately moves into the downstream group. And as soon as the downstream group has some purified material, that's moved over into formulation, so we can start taking a look at this. In each case, you have to take a look at what are the caveats of looking at using material that might not be exactly representative of the final process, and then take those risks into account in your experimental design and how you're planning out your overall development project. And then, as I mentioned previously, analytical's been working on methods throughout this process, and early on, the methods are still in development. So you also need to take into account the fact that, they may not have the exact precision they will have 3 months later. All of this has to be taken into account when you're looking at how you're going to develop your product and what risks you're willing to accept. As each stage is finished, then of course, then we have what we call a technology transfer step, and that's where we take the process, the formulation, the analytics, and then transfer that knowledge to a manufacturing site, resulting in the manufacture of a clinical trial. So it's a little more complicated than presented in the previous slides. And then sometimes things happen which cause you to have to just kick things back to an earlier step and sometimes even start over. And again, that's because this is science, and science, as much as we would like to think is nice and stable and predictive, we all know in practice there are some always some surprises. So this is a recent surprise we had. And the lesson here is thinking that one, or in this case, 6 little amino acids won't make a difference might not always hold true. So this project was actually a rapid development project. So we were moving at quite an excessive pace, and there were multiple projects going on at one time for a nanoparticle influenza vaccine, similar to the ones that Dr. Boynton talked about in his talk. So in order to meet the goal of the project that was targeting a specific clinical trial start date to take advantage of the flu season, the strategy was we would start off with a his-tagged version of the protein components of the nanoparticle for early development work. The rationale is it's quick to produce and pretty simple to purify, all things considered, because you can use affinity to use a nickel column. And then we would bridge to the non-hist version, so do some comparison studies once cell line development work is completed, which reduces the timeline by easily 2 months, if not more. And the risk assessment, when we talked about the strategy, was low. We've used this strategy before in other projects. His-tagged and non-his-tagged material perform very, very similarly in terms of any physical chemical characteristics, with the exception of the actual purification chromatography. The challenge we ended up facing was the fact that the non-his-tagged version was not stable under conditions that worked for the his-tagged material. And to kind of get a very strong example of that, take a look at the graph on the right, and that's the purity of the nanoparticle under frozen conditions at -20°C, where the material was intended to be stored. And as you can see in the orange line, we had found a formulation that worked very well for the his-tags. It's in purple in the table below, was high pH, high salt, but the material was very stable. And those conditions were still within, acceptable injectable conditions for clinical trial. So we have 10-week stability. Looked great. For the non-his-tag version, as soon as that was assembled in exactly the same way as the his-tag version, we found out that by 1 week, the entire particle had fallen apart under the same solution conditions. So what we had to do was, well, first we talked about some different storage conditions, looking at very low temperature storage, which for anyone who's followed the COVID-19 story over the last year and a half knows that that can cause some logistical challenges if you have to store it less than -60. We talked about looking at a liquid formulation. There were some other barriers to that in terms of time. It takes longer sometimes to develop those formulations. And so we ended up having to go back and redevelop both purification processes, which are also strongly affected by the pH lability of a molecule, as well as the formulation. And so what we ended up moving to was a lower pH of 6, as well as very low salt, which was much more tolerant for this molecule. And that did result in the successful manufacture of clinical drug product, which is now in trials. Another interesting thing that came up was we found out that sometimes solutions forget that equilibrium exists. And what I'm talking about here is a time when we're trying to optimize the purification process of an antibody to reduce the time for the overall process and increase the yield. And so one aspect that was being looked at was the final UFDF system. And that's, again, where we're doing a buffer exchange and a concentration step. And the strategy was to see if we could change when we added the viscosity increasing excipients. And in this case, I believe it was sucrose, moving that to the end of the process. Because what that does is it reduces the resistance to membrane permeation during the actual dialysis and concentration. And then you can just spike in the concentrated sucrose at the end. And that would reduce the operation time for this particular unit operation from greater than 24 hours to less than 9 hours, which is a huge gain in terms of the logistics of producing this at a very large plant with a lot of people involved. And there, the challenge we found out was that when we did try this, the drug substance osmolality was higher than that in the initial process, and not just a little bit higher. We were looking at 600 milliosmoles per kilogram versus 300 milliosmoles per kilogram, and that is well outside any type of assay variability and just not acceptable for the product. The theory that we ended up kind of settling on, although there's still some work being done to kind of formally prove this, is that we were most likely observing what's called Gibbs-Donnan effect. And for anyone who doesn't remember that, you do have to go back to your basic chemistry courses. And that we were seeing this occurring during the new UFDF process prior to excipient addition. And for those of you who don't remember it, what occurs here is that some ions can be selectively retained or excluded across a membrane, especially in the presence of non-permeable materials like proteins. This can manifest in pH, conductivity, or osmolality shifts, and it is highly molecule and excipient dependent, which means it's not really a predictable type of effect. Saying we've got X protein plus X excipient means we have to worry about this. Unfortunately, it's something you only see when you actually run the operation. The solution, at least at this time, is to retain the initial UFDF process step. We weren't able to find a workaround, so instead we focused on optimization of other unit operations in order to overall optimize the process, because we looked at things like clarification, chromatography, etc. We do run into a lot of different challenges, but that makes life interesting. And the important point is that at the end, once we've resolved any challenges that have come up and gone through our full development, we end up with a safe vaccine with consistent quality that's produced for human clinical trials, where we can, verify the efficacy. We move from that little Eppendorf tube with those molecules that have been designed in basic research through a full process development and manufacturing effort, ending up with a bioclinical vaccine that eventually gets tested in people and hopefully improving world health. Well, thank you very much for listening to my talk today. There are way too many people to acknowledge. So here are just a select few. First, the vaccine production program staff for the last decade. Select members of the vaccine production program influenza team across the VBP. These are across multiple groups. Our VRC leadership, the VRC project management. That team is the team that keeps us on track. And then also our pilot plant, which is under contract to Labs Biomedical. So thank you very much, and hopefully I'll be able to interact with you in future Q&A sessions.